All right, so let's just start. I'll start by introducing the speaker. Welcome everyone. Today's second day of the RBCDSAI's International Summit on Data Science and Artificial Intelligence. I'll be your host for the first session, which is between 2 to 3:30 p.m. My name is Adi N. Naganathan. I am a faculty at the Department of Biotechnology, Indian Institute of Technology, Madras, Chennai, India. Today's first speaker is Sharanya Desai, and her title of the talk is Applied Artificial Intelligence for Assessing and Improving Outcomes in Epilepsy. Just to give a brief introduction, Sharanya received her bachelor's in engineering in electrical and electronic engineering and master's in biological sciences from BITS Pilani, India, and PhD in bioengineering from Georgia Tech, USA. During her graduate studies in Dr. Steve Potter's lab at Georgia Tech and Dr. Robert Gross's lab at Emory University, she worked on a novel multi-electrode microstimulation approach for reducing seizures in a rodent model of epilepsy. She is currently a principal research scientist at NeuroPace, a medical device company that manufactures closed-loop brain-responsive neurostimulators for epilepsy. Her research focus is on applying data science techniques for understanding and improving chronic brain diseases. She is also a recipient of the International Schlumberger Faculty for the Future Fellowship and the author of publications and patents with a focus on understanding and improving human health. So, I welcome Sharanya, and also other speakers of this particular session. So, Sharanya, you can go ahead and start. You have about 25 minutes and 6 to 7 minutes for questions. Audience can post their questions via this Q&A's tab that's present. And let's keep it scientific and let's start. All right.
Okay, cool. So can you see my screen now?
Yes.
All right. So first of all, thank you organizers for inviting me to speak. And just an FYI, it's 1:30 p.m. for me, so I'm completely relying on coffee to keep me awake at this time. And another thing is this talk is of super interest to me because I grew up in Chennai. I'm a Chennai girl. So given that the audience is mostly from Chennai, I'm really excited to do this. Okay, so let's go. Here are my disclosures. I'm an employee of NeuroPace, and obviously I get paid. So those are my disclosures. In the agenda are 3 topics. So first I'll introduce the device and the types of data we capture. And then I'll talk about 2 research focus areas for NeuroPace. The first one is to assess patient outcomes, and the second one is to improve patient outcomes. Okay, so let's get started with the introduction. So at NeuroPace, we make a closed-loop brain-responsive neurostimulation device. It's called the RNS system. And this cartoon here, it shows you what the system looks like. It's implanted in the skull. So a portion of the skull that is the shape of the device is taken out, and this device is put instead of the skull. And the device can be connected to up to 2 leads. These leads can either be 2 strip leads or 2 depth leads, or a combination of strip and depth. So this device received FDA approval for use in the US for the adjunctive treatment of adults with medically refractory partial onset seizures. So this means the device is approved for adults who have not responded to seizure medications, and for those for whom we know where the seizures come from, okay? And those brain areas where the seizures come from are where the leads are targeted. So the device continuously monitors brain activity. It's sensing and sensing. And if it detects patient-specific abnormal patterns, it sends electrical stimulation to try to stop these abnormal patterns from evolving into a seizure, okay? So in addition to going through this closed cycle, the device also records snippets of brain activity. We call these electrocorticographic data, or ECOG data. And it also captures other metrics about the brain, but I'm not going to get into those details for this talk. So an ECOG file that is captured by the device, it looks like this. It typically consists of 4 channels of data. So you can see each of these lines, its squiggly line is a channel of brain data, and those are the brain waveforms in one particular patient. So each channel is sampled at 250 hertz. Until date, we've captured over 5 million of such files from over 3,000 patients, making it the world's largest ambulatory ECOG data set, okay? So in addition to capturing these ECOG files, we've also captured other information about patients, such as their seizure rates, anti-epileptic drug information, like what medications they're on, patients' clinical characteristics such as their age, gender, age of epilepsy onset, and so on. So the point of this slide is to show that we do have a rich and big data set, big brain data set. And I am one of the lucky ones to get to work on this data set. So as far as the ECOGs are concerned, the ECOG files are concerned, there are two main types of ECOG files. The first one is called the long episode ECOG file. And as the name suggests, so these ECOG file contains long episodes of abnormal brain activity. So they can either be seizures. So shown here is one example. This ECOG file contains a seizure. So that's the thing that you're seeing, the high amplitude, high frequency signal that you're seeing there is a seizure. But the long episode ECOG files can also contain long trains of other types of epileptiform events, not necessarily seizures, but long episode ECOG files are triggered whenever the brain has a long train of something abnormal going on, okay? So shown here at the bottom is just a spectrogram representation of the same signal. So this is one type of data that is captured by the device. We also capture scheduled ECOGs. So these are ECOGs that are just stored at particular times of the day. So at 2 a.m. and 2 p.m., whatever is happening in the brain, a snapshot of that is taken and it's stored in the device. So I'm going to be talking about these two types of ECOG files, okay? So this slide will kind of set the tone for the rest of the talk. What you're seeing here on the y-axis is the median percentage reduction in seizure frequency, and the x-axis is time since implant. So the data here goes from year 3 since implant to year 9 since implant, okay? And these bars are increasing in height, which shows that patients continue improving over time. And by year 9, we have a 75% median seizure reduction. This is absolutely great, especially for this patient population who have not responded to medications. But there are a couple of things I want to point out here. The first thing is that patient reports can be incomplete and inaccurate. So we know that humans are not very good at keeping reports of everyday life events, as well as, they're not very compliant. And seizures that happen in the nighttime they go unreported. So these patient reports can be incomplete and inaccurate. Also, if you look at this graph, it's taken about 3 years to achieve the 60% reduction in seizures. We think we can do much better. So think that we should be able to achieve really good seizure reductions much faster than 3 years, okay? So to just rephrase what I just said, there are a couple of unmet clinical needs. The first one is to come up with an objective assessment of patient's clinical state, so we know how a patient is doing without having to fully rely on patient reports. The other thing is an objective is to arrive at optimal clinical outcomes faster than what we have shown here. So it's taken about 3 years to achieve 60%. We want to do this faster, okay? All right, so this I have to read out to make the regulatory people in my company happy. The slides that follow are a presentation of research and include discussions of functionality not currently available for the RNS system. Okay. So the second topic is research to assess patient outcomes. So the hypothesis here is that the ECOG files that are captured can somehow tell us how a patient is doing. So maybe something in it will tell us how the patient is doing. So that will help us objectively assess how the patient is doing without having to fully rely on patient reports. So there are two types of ECOG files I discussed. The first one is scheduled ECOG. So these are captured at specific times of the day. So one of the objectives is to just look for features in these ECOG records that are correlated with patient outcomes. And the second type of ECOG record, which is the long episode type. So some of them can contain seizures and some of them don't. So the second objective is to just build a reliable seizure classifier. So to classify long episode ECOG records as either seizures or not. So then you can say that, okay, in this month, patient had X number of seizure ECOG records, and in the next month, the patient had Y number. And if X is greater than Y, then you know that the patient was not doing well in that month, okay? So these could help physicians objectively know how the patient is doing. All right, so first we'll look at the scheduled ECOGs. So we first went in and extracted like handcrafted features. So the data analysis method here is very simple. So we take the time series waveforms and we extracted features like interictal spike rate. So this is nothing but the number of these spiking events that happen in a minute or in a second, or the rate of those events within a given time window. And then other things like spectral band power, okay? So we extracted these, and we then did usual analysis, like statistical methods were applied on these features to see which ones correlated with outcomes. So here's a list of all the features that were extracted: spike rate, total spectral power, delta, theta, alpha, beta, low gamma, and high gamma. So these are like usual, very common features that are extracted in the domain of neuroscience. And then another thing we did was to focus on specific groups of patients. So MTL group of patients are patients where seizures begin from a specific part of the brain called the mesiotemporal lobe. It's a deep brain structure. So we did the analysis on the MTL patients separately and on neocortical patients separately. But in this figure, I'm just showing you the data for MTL patients. So even within the MTL patients, we went in and selected the patients who reported that they're doing really well. So those are the upper response quartile patients. And then in another category, we put all the patients who said they're not doing well. So who haven't had a very good response to stimulation, okay? And the different columns here, it shows the time since implant. So this is year 1 since implant, year 2, 3, 4, 5, 6, and 7. So in the first year that the device was implanted, you can see that the upper response quartile patients' ECOG features and lower response quartile patients' ECOG features, the distribution looks pretty much similar. But as the upper response quartile patients improve with time, and by year 7, they pretty much become seizure-free, you see these ECOG features also changing, and it reflects how the patient is doing clinically, which is really amazing. And you don't see that in the lower response quartile patients. So these patients have had pretty much the same clinical outcome, and the ECOG data also has remained somewhat same. Okay, so that was one. And when we discovered that there are ECOG features that are correlated with outcomes, we wanted to explore more. So we applied other techniques. So transfer learning is like a widely used technique in where you take a pre-trained model and you apply those models on your data and use that as a feature extractor, basically. So the data preparation here consisted of taking an ECOG channel, converting it to a spectral image, because CNNs love two-dimensional data, and using a CNN. So this CNN could be any pre-trained CNN model. So we used the GoogleNet Inception V3 model as a feature extractor and extracted a multidimensional feature vector. And then that was passed through a PCA and a TSNE step to just reduce it to one spot, which can then be visualized. So essentially what happens is time series waveform goes into this pipeline and you get a single dot on a two-dimensional plane. So when we did this for all the data captured from our lower response quartile patients, we get the points seen here in blue. And for the patients who said that they're doing really well, we get these green data points. So this just like beautifully clusters. They just cluster out so well that we were really surprised that some technique like this would apply so readily to physiological data. So now we can, okay, I'm going to keep moving this. All right. So now we can go in and take a look at data points that are in different spaces of this two-dimensional plot. So if you take a look at a few example ECOGs in this box A, there's a lot of spiking. It looks noisy. And then let's go ahead and select like box C here. So it's really calm and clean, right? And something in between, you see a little bit of spiking. So there's truly a stratification going on here. So all the noisy ECOGs are coming from patients who said they're not doing well, and all the clean ECOGs are coming from patients who reported that they're doing really well. Okay, so in case all that, what I just said didn't make sense, this one slide will kind of drive home the point. Okay. All right, so if you just focus on this, I'm just going to stop this video. Sorry, okay. If you just focus on this graph on the left, this is data from one patient. On the y-axis is disabling seizures in 28-day moving average bins. And on the x-axis is just the calendar year. So it shows you data from 2008, 2010, 12, 14, and 16. So the black plot line shows you how the patient is doing, essentially. So the patient was not doing well in the beginning, but on day 1220, there's a device programming change, which is indicated by this blue line. And after that, the patient just becomes seizure-free. No more seizures reported after that. So now let's take a look at the brain data. So here, okay, this is a really cool, all right there. So as you can see, these days are increasing. So all this data was captured before day 1220, and then I pause the video a little bit around day 1220 just to add a little bit of dramatic effect, and then this is after day 1220. So the brain signals just completely shift, okay? It just completely changes when the patient starts responding. And this is a very interactive plot, so go click on. So this was captured before the big change, and this example, which is really quiet. So you see the big difference there. So brain signals can tell you how a patient is doing clinically in epilepsy. So that is the moral of the story here. And the second objective here was to take these long episode ECOGs and reliably classify them as seizures and non-seizures, okay? So the problem with that approach is that seizures vary a lot from patient to patient. So these are 3 example patients, and if you can see, that's a seizure there. So this is another example from another patient, and that's a seizure. And these are just baseline activity. That's not a seizure. So it just varies a lot from patient to patient. So building like a feature-based algorithm or a rule-based algorithm wouldn't really work. So we wanted to train deep learning models. And the problem with training supervised deep learning models for like a binary classification problem is you need a lot of labeled data. And with a NeuroPace device, we have a lot of data, but it's not labeled. So we first needed to solve the problem of labeling these ECOG data sets, okay? So the people who are most suited are physicians. So physicians are really the people who should be labeling these data sets, and they don't have the time. So we built a tool to make that labeling process more manageable for physicians. And the way we did that is we took these time series ECOG records, we converted them to spectrograms, passed them through a pre-trained CNN, and used these CNN as feature extractors. And for each spectrogram image, we got a vector of features. So for 4-channel spectrogram images, we got 4 channels of features. We concatenated those and then used clustering algorithms in a reduced 2-dimensional feature space. The clustering algorithm was Bayesian Gaussian mixture model. We used that for clustering all the ECOGs within a patient. So when we did that, we got plots like this. So these are 6 example cluster plots from 6 example patients. So for example, if you take a look at this patient, right? There's about 10 clusters here. So there are 10 different types of ECOGs that are captured in this patient. And this particular example is a really interesting one because this patient had the maximum number of ECOGs of all the patients that we had for the labeling project. And we labeled all the ECOGs within 40 minutes. And otherwise, without a clustering tool like this, it would have taken over a month. So this really sped up the process of labeling a large data set. And once the labeling was done, it becomes a really simple problem. So you have big labeled data set, you have big computing power, just put them together and that's it. You have models which can perform your binary classification task pretty well. So the way we did the training and testing is we had 193 patients' data that was included for this study. And out of these, 113 patients' data were used for internal labeling. So we just had someone internally at NeuroPace do the labeling in 113 patients. And 80 patients was sent to an external expert. So he independently labeled data in 80 patients. So out of the 113 patients, we did an 80-20 split. So 90 patients' data were used for training as well as validation. And 23 patients' data were used for test. We trained a range of like CNN and ResNet models. And the performance of these models were tested on the 23 patients as well as the 80 patients that were independently labeled by an expert. So this really is the strength of the analysis, that we tested the model on a data set that was independently labeled. So after that was done, we did do a due diligence as far as model architectures are concerned. So we started with a really simple 6-layer shallow model, used that to come up with a baseline level of accuracy, and then we kept increasing the depth. We also experimented with ResNet 18 and 50, and the results that we get are plotted here in this precision recall curve. So these are good curves for like comparing the performance of different models. A perfect precision recall curve would make a right angle here. So as you can see, these are the different types of models we trained. And the ResNet 50, which is also the most complex model with the most number of training parameters, was the model that did the best. Okay, and these confusion matrices on the right, it also confirms the same story. So we get the best diagonal for the ResNet 50 model. So overall, we get a close to 96% class balance classification accuracy on this binary classification task. And this has not been achieved before. The main reason we got to this accuracy level is purely because of the amount of data we had. And in this, I just go on to show how the classification accuracy improves as a function of the amount of data used for training. So when we used only 10 patients' data, so that's the level of accuracy. And as the number of patients' data used for training increased, the accuracy generally goes up. And we found that it takes at least 30 patients' data to achieve a classification accuracy of greater than 90%. Okay, so now that we have a reliable binary classification classifier done, like a binary seizure classifier done, so now you can take all your long episode ECOGs, pass it through the classifier, and identify the different types of seizure ECOGs, and then plot when these seizures happened. So as you can see in this particular patient, it appears that, I'm going to push this to the side. It appears that the patient has been having more seizures in the most recent months. So this shows that, if you present a report like this to the doctor can take a look at this quickly and decide that he or she has to change the course of treatment for this patient, because whatever the treatment is right now isn't working. Okay. So that concludes part 2 of my presentation, and moving on to the final part, which is to the research that we're doing to improve patient outcomes. Okay, so like I told you before, it takes a long time to achieve 60% reduction in seizures, and we want to get to that reduction much faster. Okay, we don't want to wait for 3 years to achieve good outcomes. So one of the alternative approaches for stimulation we had in mind was, because we have this huge data set of patients that we've already treated, right? So we thought when we get a new patient, we'll find other patients in this big data set who have seizure dynamics similar to your given input patient, and then see what worked in those patients and apply the same thing in your given input patient. So that makes sense, right? So we wanted to try this out. The objective here is to search through 1 million ECOGs. So we started with 256 clinical trial patients in the search data set, and they contributed 1 million ECOGs. So we wanted to search through 1 million ECOGs quickly to identify patients with similar seizures as your given patient. Okay, and this had to be reasonably fast for it to be practically useful and also scalable, because right now we have the search data set consists of 256 patients. In the future, we'll obviously keep adding more patients to the search data set. So whatever we build had to easily scale. Okay, so the first thing we did was, so within individual patients, we went in, took all their ECOGs, we sent it through this data processing pipeline, which consisted of feature extraction and PCA to TSNE dimensionality reduction, and just plotting the ECOGs in this 2D plane. So there's a few different types of CNN models we experimented with as feature extractors. So first thing was just pre-trained models, like I've been talking about before. The other are models trained on auxiliary tasks. So we chose a task that was relevant to the problem we're trying to solve, which is basically an ECOG classification problem, and we used that as a feature extractor. The other thing is we also tried deep ranking models. So these are trained using triplet loss functions, and you send in 3 input images. The first one is an anchor, the second is something which looks like the first one, and the third is something which does not look like the first one. So you train it on triplet inputs with a triplet loss function, and you get embedding space. So the concept is basically the same. You take all your ECOGs in a given patient, all the brain recording files, and you come up with a 2D embedding space where all the similar looking ECOGs are close together. Okay, and then you apply clustering algorithms like the Bayesian Gaussian mixture model to automatically cluster all these ECOGs. So these are examples from the first 3 patients in our database, and patient 254, 255, and 256. So you see the different types of clusters that are produced in each type of patient. So this was step 1. After we did this, we selected only the centroid ECOGs in all these clusters. So the reasoning there was that the centroid ECOG will pretty much look like all the other ECOGs within the cluster. So we thought it was sufficient to just grab the centroid ECOGs for the tool that we were building. And so that brought down the search data set from 1 million ECOGs from 256 patients to 5,000 ECOGs, which represented the 1 million ECOGs, okay? So because we only grabbed the centroid ECOGs. So after that was done, the 5,000 ECOGs were taken. We applied the same techniques as before. So again, we represented the 5,000 ECOGs in a 2D space where similar looking ECOGs are close together. So what we come up with is this. So I think the first thing you'll notice is that there are 3 giant clusters. But the reason these 3 giant clusters were produced is because the most of our ECOGs are 90 seconds long. So they're 90-second long files. Some ECOGs are 180 seconds. So that resulted in this second cluster, and a few ECOGs are 45 seconds. So that was the third cluster. So that's the first thing that attracts you, that you observe in this plot. But that's really not the point of this plot. So we are really interested in these microclusters. And if you take a look at all the ECOGs within this cluster, the pink one that is kind of zoomed in here. So this is the centroid, and this is the nearest neighbor. So this is the second nearest neighbor. So you can see that the nearest neighbor to the centroid looks very similar to the centroid. And this comes from a different patient. This is a different patient, and each of these came from a different patient. Okay, so in this way, we are able to quickly identify other patients with similar seizures as your input patient. And this is just another example cluster here. So if you take a look at all the ECOGs within this pink cluster, this is a centroid, and you can see all the other ECOGs from other patients that kind of matched your centroid ECOG. So now if you have an input patient that comes in, you apply the same process. You extract, you select a seizure ECOG from your input patient, you throw it into the embedding space, and you use the K nearest neighbor approach to find similar patients. And once you find the similar patients, you make a graph like this. So the first plot here is all the frequency parameters. So when you do stimulation with a neurostimulator, there are a few stimulation parameters like the frequency of stimulation, the charge density, the path, and so on. So shown here is one such parameter. It's the frequency of stimulation. Everything shown in green here are the things that worked in similar patients, and the things shown in red are frequencies that did not work. Okay, so you compare what worked, what did not work, and you select the ones that worked. And you do that for every single parameter. So just showing you 2 parameters here. The frequency, it just appeared that the 330 hertz stimulation was pretty good. So that's a promising frequency to try in your patient. And also it looked like lower charge densities worked. So that's another promising feature to stimulation parameter to try. So that is what this kind of tool will suggest that you try in your patient. Okay, to summarize what I just said, the signal processing, machine learning, and deep learning techniques were used to discover potential objective biomarkers of epileptic patients' clinical state. We used an unsupervised, semi-supervised clustering technique to assist with expert labeling of ECOG records. And deep learning models trained to classify ECOG records as seizures and non-seizures performed really well with close to 96% classification accuracy. And we found patients with similar seizure dynamics using the student by creating 2D embedding spaces where similar looking ECOGs are close to each other. And then we used a KNN approach, K nearest neighbors approach, to identify K nearest neighbors. Also, this brings me to the most important slide of the talk, which is the acknowledgement slide. So that is my research team. This is all my teammates. These are a few of our patients. So they actually have the device implanted in them. And this is our chief medical officer. So she really knows the answer to every question you can take to her. And we also have a very good social media presence. So follow us on any of these channels. So let's see. So US FDA indication for use, read it when you have time. And I also need to show you our labeling information. That's it.
All right. Thank you, Sharanya, for the wonderful talk. There are several questions, and I believe the most common question, I think, is probably comes to the first few slides, where people are still asking questions on what is the difference between the EEG and the ECOG, and can you explain what exactly do you measure and how do you go about measuring it? That's one of the many questions I see here. It seems to be repetitive.
Sure. So EEG is a term that's commonly used for referring to data that is captured from the scalp. So when patients go in for intracranial monitoring, right? So they go to the physician's office, they get hooked up with electrodes that are put on the scalp, and those electrodes are connected to a machine that is recording signals that are recorded just on the scalp. So that is commonly called EEG. ECOG is something which is measured from within the brain. Okay, so like I showed here in this figure, the leads are actually going inside the brain. The leads are implanted in different lobes, like the temporal lobe or the neocortex. So the data that is getting captured from within the brain, that signal is usually called ECOG. And the NeuroPace device, the device that we have, it captures ECOG data, not EEG, ECOG. So it's from within the brain.
All right. So the other one of the other questions is how do you differentiate between the regular brain activity signals and seizure signals, and can you actually distinguish between when a patient is lucid and when he or she is going through this seizure from the brain
Yeah.
activity? I mean, obviously that's what you're trying to do here, but yeah, can you explain more on that?
Sure. So, like a trained physician can just look at the ECOG activity and just tell, without any doubt if it's a normal or seizure. Of course, it's not always the case. Sometimes there's confusion there too. But I'll show you this example. So this is 4-channel data. Okay, so that is one channel of data. That's a second. This is the third, and this is the fourth. So this high amplitude signal that you're seeing here, so that is a seizure. So when there's a seizure, you would see some kind of abnormal pattern in the ECOG data. So this is in one example patient, and I think I also showed a few other slides with examples from a few other patients. Okay, here. So this is a spectrogram view, so it's time series, then we applied Fourier transform, and then we made these plots. So that signal that you're seeing here, this yellow thing, which is high power data, so that is a seizure in this patient. And another patient, the seizure can look completely different. So that's a seizure here. And in the third patient, there you go. So that's the seizure. So it is very patient specific. Like a seizure in one patient might not look like seizures in another patient. And also seizures in one patient might look like baseline in another patient. So yeah, but a trained physician will be able to pick these differences out pretty easily.
All right. So any questions from the panelists? All right. If not, Sharanya, you can answer the questions that are put up by the attendees. You're not obliged, but you can still try to answer many of the questions if possible. All right. So thanks, Sharanya, again for the wonderful talk.